to move on. So we're still in the same unit. So this unit that we're in right now is called cells. We started off talking about microscopes. Then we actually went into the cells, cell organelles, those sorts of things. Now we're going to finish up this unit by talking about what we call cell processes. Basically, things that happen, processes that take place within the cell. We're going to talk about 4 main processes. Osmosis and diffusion we'll talk about together over the next week or so classes. It'll take us after Thanksgiving. Now we'll talk about photosynthesis and respiration separately. So we're still in the same unit. And now we're just continuing on. Brandon?
I have no idea why the salt makes you thirsty.
Oh, you have a new idea? What's your new idea?
That the salt breaks down the water, it breaks down the compounds, and then.
Okay. Almost like I said, we'll learn towards the end of class today exactly why salt makes you thirsty. All right. So when we start off here, and I know from what other classes said that last year you guys spent a lot of time or some time on solutions.
Yeah.
So we're going to go real quickly through today because I think most of this you probably remember from last year. But we need to review solutions and concentration before we can actually start to talk about osmosis and diffusion. So we'll go kind of quickly today. So if we think about concentration, okay, we'll use Kool-Aid as an example here. If we have 2 glasses of Kool-Aid, A and B, which one is more concentrated for Kool-Aid? So you see? Yeah, B is. Has a stronger taste. Which one would have a higher concentration of water? Right? Sample A. It would taste weaker, be a more watery taste. So concentration is sort of, you can think of it as sort of the percentage of different materials. No, glass B maybe is 20% Kool-Aid, 80% water. Glass A maybe is 5% Kool-Aid, 95% water. Right? It's not the same as the amount, and we'll get to that in a minute. So if I were to draw a picture showing what this actually looks like, what do the molecules look like? They're the same amount of liquids, but in sample A, there's a lower concentration of those Kool-Aid molecules. Sample B has a higher concentration. They're more crowded. All right, so we have some terms that we use when we're talking about solutions. So how could you make your Kool-Aid a more concentrated Kool-Aid solution? You do 2 things. Juliet, what's one? You could add more Kool-Aid to your solution, more of the powder, or drink.
Take away some of the liquid.
You could use less water. Either one is going to give you a more concentrated Kool-Aid solution. The term we use sort of for the opposite is a dilute solution. How can you make this a more dilute Kool-Aid glass? Kayla?
Add more water.
You could add more water to it. Or you could have made it, Brandon, with less of the Kool-Aid, either one. Okay, so Kool-Aid is what we call a solution. A solution has 2 parts to it. So if I have some water here, I have some salt. Okay? I can put some salt in this water, and I'm making a solution. The 2 parts of a solution are called the solute and the solvent. What was the salt? I mean,
Solute.
that's the solute, the thing that seems to disappear, the thing that you're adding. And the solvent in this case was what?
Water.
The water. Now, what's actually happening here? So I don't see the salt anymore. Is it gone?
No.
No, what's actually going on here as I dissolve the salt in the water, Tom?
Dissolving.
Yeah, but what does that actually mean?
It's breaking down in the
Oh, okay. So yeah, it's getting broken down into basically smaller pieces. Okay, when salt is in this form, salt crystals, pretty large, you could see them, okay, just by looking at it. You can take one individual salt crystal and you can actually see it. Okay, it's pretty small, but you can see it. When you dissolve something, basically that salt crystal gets broken into smaller pieces and smaller pieces until it's broken into individual salt molecules, which are very, very tiny, okay, beyond microscopic. And so they're still here, they're still in there, okay, they're still salt, but they're in such small pieces that you no longer can see them. Okay? If I tasted this water, would it taste salty?
Yeah.
Yeah, so we know that they're still there. They're just invisible. They're broken down so small that we cannot see them. Could I add an unlimited amount of salt to that?
No.
No. Eventually, the water will be holding all of the salt that it possibly can. Do you remember the word for that?
Saturated?
Yes, it's saturated solution. It's holding as much salt as possible. Good. All right, your 6th grade teachers would be proud. All right, so in this Kool-Aid example, what is the solute in the Kool-Aid? Matt?
Kool-Aid powder.
Kool-Aid powder. Mainly it's sugar, some flavoring and coloring. And the solvent is water. In our case, we're almost always going to use water as a solvent, but there are other solvents as well. All right, if we look at some examples of a few solutions here. Sample A and B here, of these O molecules. What's the solute in that example? Right? The O's. And the solvent in this case is what? Water. Which has a higher concentration of O's? Rosemary? B does. And which has a higher concentration of water? Kimberly? What's that? A. All right. Another example. Now we have salt molecules. That's what these represent. What is the solute? Peter?
Salt.
The salt. What is the solvent? Juliet? Water. Which is a higher concentration of salt molecules? Linda?
Sample A.
Sample A. Which has a higher concentration of water? Kayla?
Sample B.
Sample B. Okay, one more. Now we have glucose molecules. What's the solute? Brandon? Glucose. What's the solvent? Pradeep?
Water.
Water. Which has a higher concentration of glucose? Caitlin? Sample B. But there's 6 molecules in each. Okay. So this is when we say that concentration and amount are not the same. Because we have the same amount of glucose in sample A and sample B. Well, like Caitlin just said, but there's different amounts of water, which means the concentration is different. So yes, Caitlin's right. Sample B is a greater concentration of glucose, which has a greater concentration of water. See? Sample A. So what's the difference between these two beakers? Matt?
That they have different amounts of water.
There's different amounts of water. Same amount of glucose, but different amounts of water. All right, so now we talked a little bit about concentration. We can start to get into actually diffusion. So the cell membrane is important, and what did we say the function of the cell membrane is what? Or one of the functions, because there's multiple. Nicholas?
To allow things in and out.
Yeah, to control what goes into and out of the cell. And that's related to diffusion. Okay? The cell membrane knows the thin layer around the outside that allows molecules in and out. And when we talk about membranes, we have some terms that we use. We could say that the membrane is impermeable, permeable, semi-permeable. Anyone want to guess what any one of those words means? What is, well, that's impermeable. I want to take a guess as to what that means. Impermeable. Right?
I think that the cell membrane doesn't want to cave in.
Yeah, it means that substances cannot pass through. Impermeable. What do you think permeable means?
Things you go in and out.
Yeah, that things can freely pass through. Leaving us with semi-permeable. Brandon?
Only like certain things can pass.
That's right. Some things can pass through, but not others. What do you think the cell membrane, which category would it fit into? Abhi?
Oh, semi-permeable.
Yeah, it's a semi-permeable membrane. It's permeable to certain molecules, but impermeable to others. It's a semi-permeable membrane. Cell membrane, it looks like it's actually a double layer. It's called a phospholipid bilayer. Okay? Which means it's sort of two layers. The phospho part are these round circular parts. They face outside and inside. And then in the middle are these tails. Those are the lipids. Phospholipid bilayer. Also, sort of stuck throughout the cell membrane are these proteins. That's what these purple and orange things are. And they have different jobs to do. Some help the cell communicate. Some help substances go through the membrane. They have lots of different jobs. If we look at sort of another view of it, this is the cell membrane. This is the two layers. Some substances that are very small can just sort of go through the cell membrane like it's not even there. The cell membrane is permeable to things like water, oxygen, carbon dioxide. Those are generally small molecules that just sort of slide through the cell membrane and can go from the outside to the inside very easily, or the other way around. It's like it's not even there almost. But larger molecules, proteins and sugars and starches and carbohydrates, they're too big to just slide right through. They often have to go through one of those proteins to get into the inside of the cell. All right, so if you think back to our Kool-Aid example. Concentration. Think of it as the strength of a solution. So if we look at this Kool-Aid on the far left, what does it have a low concentration of? Peter?
Kool-Aid.
Yeah, what does it have a high concentration of?
Water.
Yes. If we look at the far side, how could you describe that one? Julia? Yes, there's a high concentration of Kool-Aid, but a lower concentration of water. You know, this one's maybe 30% Kool-Aid, 70% water. This one's maybe like 5% Kool-Aid and 95% water. Which one would you drink if you had your choice?
Second to the left.
Middle one?
Second to the right.
The last one, this one?
Second to last.
Too sweet.
I picked one in the middle. You have middle one. Second to last.
Middle one.
Alright. Now let's finally talk about diffusion. So, first off, we keep talking about molecules. We'll be talking about molecules throughout this section of the unit. What is a molecule? Right?
Something that can be breaking down into.
Okay, sort of. It actually could be broken down farther, but it's then going to be a different substance. You know, for example, salt. If I had these salt crystals, if I were able to like break one salt crystal in half, would I still have salt in each of my hands? Yes. Then I broke those down farther, farther and farther. Eventually, if you could do this, you'd get to a single molecule of salt. That is the smallest piece of salt that you could ever have. If I took that one molecule of salt and broke it up any further, then it's not salt anymore. It's something different. Actually, two different things. So it's basically the molecules are the building blocks of matter. And diffusion has to do with how molecules move. So the definition of diffusion, which you're going to have to know, is movement of molecules from an area of high concentration to an area of low concentration. Molecules that are tightly packed, that are in a high concentration, will tend to spread out if possible. Sometimes I use the analogy, if I think about molecules like 7th graders, if I took every 7th grade student in our school and put them all in this one classroom, I could probably fit all of them. It'd be really tight and hot and sweaty and smelly, but I could probably fit every 7th grader in this room. Would that be a high concentration of 7th graders?
Yes.
Yes. All right, so after a minute, you'd be annoyed. But then, let's say, without really doing anything, I open up the door to Miss Bean's room. What would happen? Rosemary? People go over there. Would everybody go over there? No. How many do you think would go over there?
Half. Anyone near the door?
About half, probably. Would I have to say, okay, guys, let's go over there? Or would it just happen on its own?
It happens.
Just happen on its own. I wouldn't have to make people go over there. Then I open up the door into Mr. Arcuri's room. What happens then?
Another half closer.
Yeah, then we, again, spread out to accommodate that space. Then I open the door into the hallway. Again, we spread out further. So, molecules act in a similar way. When they're in a tightly packed area, they tend to spread out if possible. And that's what diffusion is. Molecules go from a high to a low concentration until what? When do they stop? Well, when they become equal, when the concentration becomes equal. So if I look, for example, if you have some molecules tightly packed, let's say in this room, like this happens every morning. In the morning when Mr. Arcuri comes in, he's usually here before me, he opens up that door in that corner of the room, and that's when he puts on his perfume and get ready for the day. So he sprays it in that corner of the room, but then when I walk in the morning, after I get here, I open up the door, and I can smell it all the way over here. So how come I can smell it all the way over here? So you see?
It's spread throughout the
Yeah, those molecules, when he sprayed his perfume, that little plunger bottle thing, there was a tightly packed little clump of those perfume molecules. But they diffuse throughout the room. You can actually smell it on the opposite side of the room. And that's, well, you know, it's whatever. He likes his perfume, so that's okay. Dye diffusing through water does something similar. If I have this beaker of water here, if I put a couple drops of this food coloring in, The food coloring starts off with what kind of concentration? High. It's a little clump of those dye molecules, right? This is a high concentration. And so over time, what's happening? It's spreading out. It's diffusing throughout that water. Okay? And so that's what happens in metals. They diffuse to spread out. And eventually, what is this beaker going to look like?
Green.
Just uniformly green because the molecules are spread out so there's equal concentration throughout that water. Okay, 2 more slides here. So osmosis then. So diffusion is when molecules move from high to low concentration on their own without any energy having to actually move them. Osmosis is just one special type of diffusion. And that's osmosis when it's water that's moving. So water also will go from high to low concentration. When it's water moving from high to low through a membrane, we call it osmosis. That's another definition you'll definitely need to know. The diffusion of water across a semi-permeable membrane. So if I look at this apparatus here, I actually have this tube thing. This never really works well when I try to demonstrate it in real life. But let's say I have, this is a U-shaped tube, and it's split in the middle. It's actually in 2 pieces. And you could put in that piece in the middle, you could put a membrane, a piece of plastic that's semi-permeable. It allows small molecules through, like water, but larger molecules can't get through the tiny holes in that piece of plastic. All right, so I have my U-shaped tube. There's a membrane separating it, okay, a semi-permeable membrane. And on one side, I have 2 types of starch solution, starch and water. The purple represents the starch molecule. So I put one type of starch solution on the left side, then I put another type on the right side. So what do you think would happen in this setup?
It would all evenly spread out.
What would?
Solute.
Well, the solute, let's talk about the starch molecules. Would they equally spread out?
No.
Why not?
Because it's too big.
They can't. Okay? If this membrane were permeable to starch, they would diffuse through equally, but they're not. So the starch molecules that are here are stuck on this side. The ones that are here are stuck on that side. All right, so we know the starch is not going to move. But what can move? The water. Which way is it going to move? Peter?
Right?
To the right. Where is there a higher concentration of water? On the left. There's a lower concentration on the right. We know water, like other molecules, will go from a high concentration to a low concentration. After a little while, it would look like this. Water has moved over to the right because of osmosis.
You say that
It never works very well. It always ends up leaking. The membrane ends up leaking.
Do you want us to draw this?
No, that's okay. I just want you to understand. All right, this is our last slide here. This is where we can learn why salty foods make us thirsty. So this represents some red blood cells. That's what these red things are. And they were placed in different solutions of water and salt. You know, a cell is probably about 97% water. A lot of water in a cell. All right, so let's say in A, I place it in some water and salt that's also 97% water, 3% salt. What would happen? Right? Nothing really. There's equal concentrations of water in the cell and in the liquid. So basically water molecules don't move in either direction. They actually do in the same amount. Okay, so that makes sense, I think. Now in B, I have my 97% water cell, but I put it in very salty water. 90% water, 10% salt. What's going to happen in that situation? Brandon?
The salt is gonna move into the blood cells,
Well, let's say it's impermeable to salt.
but the water's gonna move out.
Yeah, there was a higher concentration of water in the cell, a lower concentration in the solution. The cells are going to lose water, and they're going to shrivel up. This is what happens when you eat salty foods. You eat a bag of potato chips with a lot of salt. That salt dissolves in your saliva in your mouth, lowering the concentration of water. The cells in your cheeks and tongue have a higher concentration of water. Therefore, what happens? The cells in your cheeks have a higher concentration of water than the salty stuff you just chewed up. So which direction does the water go? Huh?
It goes out and it's dry out.
Out of your cells into the space around them, causing the cells in your cheeks and your tongue to shrivel up, okay? Making you thirsty. What happens when you drink a big glass of water then? Where is there a high concentration of water? Not in your cells now. In your mouth, in that swig of water you just took. And so there's a higher concentration of water, lower concentration in your cells. So which way does water go? Back into your cells, causing them to swell back up and return to their normal shape. And that's why you feel better after you drink them. The last section, if we place cells in pure 100% water, which way does the water go? Somebody else. This can't be the Brandon Holmes show today. Kaylin? Into the cells, causing them to swell up, possibly even bursting.
What?
The cells can burst. That's why if you ever